Parrotfish, a vibrant and ecologically significant group of marine fish primarily found in tropical and subtropical waters, exhibit a fascinating behavior where many species secrete a mucus cocoon around themselves at night. This behavior serves as a protective mechanism, primarily to mask their scent from predators. Below is a detailed explanation of this process, its purpose, and the biological mechanisms involved. The mucus cocoon, purpose and function. The primary function of the mucus cocoon is to provide a protective barrier that reduces the risk of predation during the night when parrotfish are resting. Many parrotfish species are diurnal, meaning they are active during the day and rest at night, often wedging themselves into crevices or hiding among corals. During this vulnerable resting period, they are more susceptible to nocturnal predators such as moray eels, sharks, and other carnivorous fish that rely heavily on their sense of smell to locate prey. The mucus cocoon serves multiple purposes. Scent masking. The cocoon is believed to trap or mask the chemical cues, scents, that the parrotfish might emit, which could otherwise attract olfactory-sensitive predators. By enclosing themselves in a mucus layer, parrotfish reduce the dispersal of these chemical signals into the surrounding water. Physical barrier. The cocoon acts as a physical shield, potentially deterring small predators or parasites from directly accessing the fish's body, camouflage and protection. The translucent nature of the cocoon blends with the surrounding environment, making it harder for predators to visually detect the fish. It may also protect against minor abrasions or irritants in the water. How the cocoon is formed. The formation of the mucus cocoon is a remarkable physiological process that involves specialized cells in the parrotfish's skin. Here's a step-by-step explanation of how it occurs. Mucus secretion. Parrotfish possess specialized goblet cells, mucus-producing cells, in their skin, particularly around the head and mouth region. These cells are responsible for producing the mucus that forms the cocoon. As night approaches and the parrotfish prepares to rest, it triggers the secretion of this mucus. The process is likely regulated by the fish's circadian rhythm, which cues the physiological response to nighttime conditions. Cocoon construction. The parrotfish begins secreting mucus from its mouth and gill areas. It uses its buccal mouth cavity to release the mucus, which is expelled in a controlled manner to form a thin, gelatinous layer around its body. The fish may swim slowly or remain stationary in a crevice while releasing the mucus, allowing it to envelop its entire body. The mucus is lightweight and buoyant, forming a loose, transparent sac that surrounds the fish without restricting its ability to breathe. The cocoon is not a rigid structure, but rather a flexible, gelatinous envelope that conforms to the fish's body and the surrounding environment, such as coral crevices or rocky substrates. Stabilization and hardening. Once secreted, the mucus begins to solidify slightly in the water, forming a cohesive but permeable membrane. This membrane is porous enough to allow water and oxygen to pass through, ensuring the fish can respire while enclosed. The cocoon typically takes a few minutes to form fully, with some species completing the process in as little as 2 to 3 minutes. The entire cocoon may take 30 to 60 minutes to reach its final form, depending on the species and environmental conditions. Resting within the cocoon. Once the cocoon is complete, the parrotfish settles into a resting state, often wedged into a crevice or nestled among corals. The cocoon remains intact throughout the night, providing continuous protection. The fish remains relatively motionless during this time, conserving energy and minimizing the release of chemical cues that could attract predators. Biological and ecological context species. Variation. Not all parrotfish species produce mucus cocoons. This behavior is most commonly observed in species within the family Scaridae, such as those in the genera Scarus and Chlorurus. For example, the stoplight parrotfish, Scarus virid, and the queen parrotfish, Scarus vetula, are known to create cocoons. while some other species may rely on alternative strategies like hiding in complex reef structures. Energy investment. Producing the mucus cocoon is energetically costly, as it requires the fish to synthesize and secrete a significant amount of mucus. However, the survival benefit of avoiding predation likely outweighs this cost, as the behavior has evolved and persisted in many parrotfish species. Ecological role. Parrotfish are critical to coral reef ecosystems, as they graze on algae, helping to maintain the balance between coral and algal growth. The mucus cocoon allows them to survive and continue this ecological role by reducing predation pressure, sensory and predatory dynamics. The mucus cocoon's ability to mask scent is particularly effective against predators that rely on olfaction. For example, moray eels. These nocturnal predators have an acute sense of smell and are known to hunt parrotfish. The cocoon likely reduces the diffusion of amino acids, proteins, or other chemical signals that eels use to detect prey. Sharks. Some reef-dwelling sharks, such as nurse sharks, also use scent to locate prey. The cocoon may interfere with their ability to pinpoint the parrotfish's location. Parasites. The cocoon may also deter parasitic isopods or other organisms that could attach to the fish during the night. Additional benefits and hypotheses. While scent masking is the primary hypothesized function, researchers have proposed additional benefits. UV protection. In shallow waters, the cocoon might offer some protection from ultraviolet radiation during early morning hours when the fish is still enclosed. Parasite deterrence. The mucus may contain antimicrobial properties that discourage parasitic infections. Thermoregulation or osmoregulation. The cocoon could help stabilize the fish's microenvironment. Though this is less studied, scientific observations, the behavior was first documented in the 1950s by marine biologist John E. Randall and others who observed parrotfish in their natural habitats. Studies have since confirmed that the cocoon is a mucus-based structure, distinct from other fish secretions. Research suggests that the cocoon is most effective in environments with high predator density, such as coral reefs, where parrotfish are a common target. The cocoon dissolves or is shed in the morning when the parrotfish becomes active again, leaving little trace of its presence. Conclusion. The mucus cocoon of parrotfish is a remarkable adaptation that showcases the intricate balance between survival strategies and environmental pressures in coral reef ecosystems. By secreting a mucus envelope around themselves at night, parrotfish create a protective barrier that masks their scent, deters predators, and enhances their chances of survival during their vulnerable resting period. This behavior highlights the evolutionary ingenuity of marine organisms and underscores the complex interplay of physiology and behavior and ecology in the underwater world.